the Chinese government, and he's giving us a tutorial. Few minutes, we will finish that. Thank you so much. Please start.
Sergey Petoukhov, cyclic physiology, cyclic codes, and algebra logic peculiarities of the genetic code system, quantum bioinformatics, and ancient Indian philosophy. This talk contains the following parts. Firstly, organisms as a set of inherited physiological cycles. Second, organisms as cyclically coded cyclic entities connected with cyclic gray codes. Third, DNA alphabets and genetic Carnot matrices related to Boolean algebra of logic. Fourth, genetic matrices of hydrogen bonds and their relation to musical harmony and universal rules of probabilities in genomic DNAs. 5. Bioinformatics, bioantenna arrays, and evolution. 6. Collective algebra logical consciousness. 7. Ancient Indian philosophy, genetic informatics, and chess. The creators of quantum mechanics, Jordan and Schrödinger, pointed out the key difference between living bodies and inanimate ones. Inanimate objects are controlled by the average random movement of their millions of particles, and the movement of individual particles is not significant for the whole object. On the contrary, in a living organism, selected genetic molecules have a dictatorial influence on the entire organism due to quantum enhancement. See the history of quantum biology. This talk presents the results of the author's research on the structural relationship of the genetic system with cyclic and algebra logical features of genetically inherited physiological systems. Any living organism is a huge chorus of genetically inherited cyclic processes. Since ancient times, chronomedicine has believed that all diseases are the result of a violation of this coordination. Our proteins are involved in continuous life-death cycles of their assembling and disassembling into amino acids. For example, the half-life of the hormone insulin is about 8 minutes. In other words, genetically inherited parts of our body are constantly dying and reborn. Considering such phenomena, physiologist Gurevich claimed the main problem in biology is maintaining shape while constantly renewing the substrate. The energy costs for these cyclic events are taken from the universal source of energy for all biochemical processes of all organisms on Earth, ATP. The lifetime of one ATP molecule in humans is less than 1 minute. During the day, one ATP molecule goes through an average of 3,000 cycles of resynthesis. Our body synthesizes about 40 kilograms of ATP per day. Another illustrative example of cyclic organization is the metamorphosis of butterflies, consisting of the stages butterfly, egg, caterpillar, pupa, butterfly. It is characteristic that no one teaches the butterfly how to get out of the pupa and begin to fly, but it gets out and begins to fly due to inherited cyclic movements of its big wings. Everything necessary for this is already available in its genomic code informatics. Many such facts suggest that the genetic coding system ensures the inheritance of coded cyclic processes, since this system itself is based on some cyclic codes. But which of the many types of cycle codes corresponds to the structural organization of the genetic code system? I have obtained some pieces of evidence about the important role of the family of cyclic gray codes for the modeling of the genetic coding system. Received results allow me to introduce the following effective model postulate. Living organisms are cyclically coded cyclic entities. A binary gray code of order n is a sequence of 2 in n power n bit numbers in which any two adjacent numbers differ by exactly 1 digit. The Hamming distance between them is 1. Gray codes of different orders form a hierarchical family. Gray codes are widely used in technology. Numbers in the gray code are fundamentally different from binary numbers, although they also consist of sequences of zeros and ones. For example, the decimal number 3 in binary notation is 011, but in gray code it is 010. In DNA, the alphabet of 4 nucleotides, T, A, C, and G, is the carrier of a system of binary positional characteristics. Firstly, 2 of them nucleotides are purines, A and G, which have 2 rings in the molecule, and the other 2, C and T, are pyrimidines, containing 1 ring, which gives a representation in blue. C and T is 0, A and G is 1. Secondly, 2 of these nucleotides are keto molecules, T and G, and the other 2, C and A, are amino molecules, which gives a representation in red. C and A is 0, T and G is 1. For this reason, DNA alphabets of 4 letters, 16 duplets and 64 triplets can be presented in the form of square tables, the columns of which are numbered with their positional indicators, pyrimidine or purine in blue, and the rows with their position indicators, amino or keto, in red. In these genetic tables, rows and columns are numbered by ordered numbers of n-bit gray codes, similar to Carnot maps, a well-known method for simplifying Boolean algebra expressions. Such numeration was applied specially to search a possible communication of the genetic code system with Boolean algebra of logic. In such tables, all monoplets, duplets, and triplets automatically occupied a strictly individual place. Each cell in these matrices, as well as the n-plet in it, is numbered by concatenation of the gray code numberings of its row and columns. We conventionally call such tables genetic Carnot maps. Arrangement of 4 monoplets, 16 duplets, and 64 triplets is shown here in matrices with numbering of columns and rows according to gray codes. How are the amino acids and stop codons they encode located in a matrix of 64 triplets constructed in this way? This arrangement cannot be predicted, since the amino acids and nucleotides ACGT are completely different in structure. The number of options for the arrangement of amino acids with their repetitions to fill the entire 8 and 8 matrix is immeasurable, much more than 10 in 100 power. For comparison in physics, The lifetime of the universe is estimated as 10 in power 17 seconds. Will this arrangement be chaotic, or will it suddenly turn out to be naturally symmetrical? The following figure shows the case of the vertebrate mitochondrial genetic code, which is considered to be the most ancient and symmetrical among the genetic code dialects. It turns out that from the ocean of possibilities, nature chose a regular and symmetric variant of the repetition and arrangement of amino acids and stop codons in this matrix of 64 triplets. The shown matrix of encoded amino acids and stop codons consists of pairs of adjacent rows 0, 1, 2, 3, 4, 5, 6, 7, identical in composition of amino acids and stop codons, shown in color here. For example, rows 0 and 1 contain the same composition and arrangement of the red amino acids, proline, glutamine, histidine, etc. Cells in Carnot maps are known as min terms, while each cell value represents the corresponding output value of the Boolean function. The presented genetic facts show one of the examples of a connection of the DNA and place alphabets with a system of Boolean functions. Mutual replacement of binary symbols, symbols 0 and 1 in the gray code, complementarity operation, generates a complementary cyclic sequence with a shifted order of terms and a unit Hamming distance between neighboring terms. The applications of this operation of complementarity to a new sequence generates the original gray code associated with the property of self-duplication and multiplication of DNA. Slide. Based on analogies between the structural properties of the genetic code system and gray codes, I am developing a family of models of cyclic genetic biomechanics using symmetry principles and my model postulate. Living organisms are cyclically coded cyclic entities. In particular, in these models, the well-known fact is used that gray codes are closely related to the fractal Hilbert curve, which allows you to discretize any space, creating a convention coordinate system in it. This relates to the problem of how genetic information recorded on one-dimensional DNA traits determines the three-dimensional morphology of living bodies. It is important that the spatial packaging of chromatin in the genome turns out to correspond to the Hilbert curve, which is its polymer fractal 3D globule, features on the cover of the journal Science. One should add else that gray codes are closely connected with Walsh functions, sequence analysis by Harmuth, and spectral logic, which are widely used in communication and other technologies. Logical holography on Walsh functions, Walsh antennas, dyadic log, and many other examples. These connections are useful for developing new approaches to model inherited physiological and genetic phenomena and to develop new artificial intelligent constructions. Along this path, data are received that the DNA nucleotide sequences encode not only the sequences of amino acids in proteins, but also inherited logical connections, genealogical coding. These data indicate that the genetic code system has two different types of code. Firstly, the well-known triplet code of amino acid sequences. Secondly, the n-plets code of the Boolean functions stated by me. Accounting for the genetic Boolean code allows you to understand more deep the innate ability of the butterfly to get out of its pupa and begin to fly, which is impossible without the logical coordination of millions of its neurons and muscle units. It is known that without mathematical logic, discussions about whether a machine can think lose all ground, and the urgent problem of creating artificial intelligence become meaningless. The presented identification of structural links between the genetic code system and the algebra of logic brings to mind the founder of mathematical logic, George Boole, and his book, An Investigation of the Laws of Thought, on which the mathematical theories of logic and probability are based. The mathematical logic proposed by Boole was considered useless for practice for decades, but now computer technologies, the internet, devices of artificial intelligence, etc., are based on it. The future of algebraic biology, in our opinion, is also connected with Boole's creation. The next section, nucleotides, hydrogen bonds, and genetic Carnot matrices. Hydrogen bonds are the most important type of chemical bonds for living things. Hydrogen bonds determine the formation of cell membranes, helicity of biostructures, and many physiological processes. Hydrogen bonds determine many properties of water, which is an important component of all biobodies. The biological importance of the properties of hydrogen bond systems is illustrated with the example of jellyfish. They consist of 98% water. Figuratively speaking, they are bags of hydrogen bonds, but they represent the most ancient species of multicellular animals with the richest evolutionary diversity and abundance of vital functions. In DNA, two complementary nucleotides A and T possess two hydrogen bonds each, and the other two complementary nucleotides C and G possess three hydrogen bonds each. This gives the hydrogen representation of nucleotides. A in T is 2 and C and G is 3. Let us consider the family of mentioned genetic Carnot matrices of nucleotides, where each of nucleotides is replaced by its number of hydrogen bonds, and each of n-plates is replaced by product of hydrogen bonds of its nucleotides. For example, triplet CAG is represented as 3 multiple 2 multiple 3. The ratio 2 is known in the theory of musical harmony under the name of the quint or the fifth. The Pythagorean musical systems called fifths are built on the ratio of the fifths, 3/2. In ancient China, the numbers 2 and 3 were considered the numbers of Yoth and Heaven, as well the feminine yin and masculine yang numbers respectively. The ancient Greeks, following the ancient Chinese, attributed a key role in the structure of the world to musical harmony and considered 3 and 2 as masculine and feminine numbers, the interaction of which generates musical tones. In general, any tuning based on the ratio of the fifths 3/2 and the octave 2/1 is called Pythagorean. Examples are the pentatonic, tetratonic, and tritonic tunings. It is known that these fifth tunings were used by different peoples all over the world long before Pythagoras, without any knowledge of mathematical laws due to an innate attraction to this tuning. See, the above described genetic matrices Q1 and Q2 of nucleotide hydrogen bonds are called quint matrices, since the ratio between the numbers of any two cells in each matrix is equal to the quint 3/2 to the integer power. It is a similar way numerical quinton matrices of hydrogen and plates for N is 3, 4, 5 are formed from the nucleotide Carnot matrices. They are used for the matrix construction of multi-stage Pythagorean quint systems, revealing the structural connection of the molecular genetic system with the innate attraction to quint systems of people around the world over thousands of years. Goethe claimed music is something entirely innate, visceral. Leibniz declared that music is arithmetic of soul which computes without being aware of it. The quint-Carnot matrices of binary hydrogen bonds are surprisingly related to the famous golden section, phi. Taking the square root of the genetic quint matrices produces golden Carnot matrices, all members of which are with absolute accuracy golden section phi to integer powers. The golden section divine proportion phi has been a mathematical system symbol of self-reproduction for many centuries. Leonardo da Vinci, Kepler, etc. It has long been used in the aesthetics of proportions, theory of coding, and optimization. The golden section proportion is noted by many authors in various physiological systems, cardiovascular, respiratory, electrical activity of the brain, etc. The discovery of a matrix connection between the golden section and genetic matrices testifies in favor of the molecular genetic roots of this physiological phenomena. From an algebraic point of view, these genetic Carnot matrices of hydrogen bonds are matrix representations of multidimensional hyperbolic or double numbers widely used in theoretical physics, computer science, etc. But hyperbolic numbers are also important for algebraic biology, which we illustrate with an example of the basic cyclophysiological Weber-Fechner law. This law speaks of the universality of algebraic principles of sensory information processing. Our sense organs, hearing, sight, touch, smell, taste, etc., differ from each other in their biochemical, cellular, and structural implementation. But the work of all these very different sense organs is subject to one and the same Weber-Fechner law. The sensation is proportional to the logarithm of the stimulus strength, where the rho is the perception threshold, K is the proportionality coefficient for the given channel. In techniques, this law has led to the logarithmic scale of decibels for sound power, But the geometric theory of natural logarithms is based on hyperbolic rotations given by two-dimensional hyperbolic numbers, For this reason, the natural logarithms at the beginning of their history were called hyperbolic logarithms, since they correspond to areas under the hyperbola. The mentioned matrix algebraic features of the genetic code system gives appropriate tools for modeling the logarithmic law of Weber-Fechner. The Weber-Fechner logarithmic law relates not only to nervous system, which arose very recently in biological evolution. This genetically inherited law is applicable to hemerotropic, heliotropic, and geotropic movements of bacteria and fungi and firm enterozoids, molluscs, and planar rodents. Weber-Fechner's law, therefore, is not the law of the nervous system and centers, but the law of protoplasm in general and its ability to respond to irritations. This citation. Next, hydrogen bonds in the statistics of genomic DNAs. It turns out that the distribution of nucleotides with 2 and 3 hydrogen bonds in single-stranded genomic DNAs of higher and lower organisms obeys the universal rule of the dichotomous fractal of probabilities. Let us explain this. Let us imagine such a genomic sequence of 4 nucleotides, CGLT, is fragmented shown here in the figure from GenBank as a binary sequence in which nucleotides C and G endowed with 3 hydrogen bonds are designated by the symbol 3, and nucleotides N and T endowed with 2 hydrogen bonds are designated by the symbol 2. Then, for example, the sequence TCAGGTA will turn into the binary sequence 2, 3, 2, 3, 3, 2, 2. Under statistical analysis, such genomic binary sequence is first considered as a text of single symbols, 3 and 2, with computer calculation of the percentage probability of each of these 2 symbols, 3 and 2. Then the same binary sequence is considered as a text of binary duplets, 3, 3, 3, 2, 2, 3, 2, 2, with calculation of the probability of each of these 4 types of binary duplets. Then, similarly, the same binary sequence is represented as text of triplets, tetraplets, etc., with each time calculating the percentages of each of the 64 types of binary triplets, each of the 256 types of binary tetraplets, etc. Thus, the nucleotide sequence of each genomic DNA is considered as a multi-layered system of multitude of templates of hydrogen bonds. It turns out that these probabilities of templates in different template texts are unexpectedly interconnected and form a dichotomous fractal tree of probabilities of hydrogen bonds. The rule of dichotomous fractal trees of probabilities of templates of hydrogen bonds is in genomic single-stranded DNAs, the probability of each template in the appropriate template text is practically equal to the sum of the probabilities of 2 n+1 plates in the n+1 text, whose denotations differ from the denotation of the considered templates by added suffixes 3 and 2. For example, in shown figure for probabilities of templates of DNA of human chromosome number 1, the probability of the duplet 3, 2 is coincides with the sum of the probabilities of two triplets, 3 to 2 and 3 to 3. See slide. This and other discovered and published universal rules or archetypes of statistical organization of nucleotide sequences in genomic DNAs are valid for all genomic DNAs already studied in our laboratory. Firstly, all 24 human chromosomes, all chromosomes of Drosophila, mice, worm, and many plants, 19 genomes of bacteria and archaea, many extremophiles living in extreme conditions, for example, radiation with a level 1,000 times higher than that lethal to humans. These universal rules in genomic DNAs indicate the existence of non-trivial algebraic invariants of a globally genomic nature, which remain unchanged over millions of years of biological evolution, during which millions of species of organisms die off and new ones appear, although locally genomic sequences are modified by mutations, natural selection, mechanisms, etc. Thus, a Jordan prediction about the existence of laws of life missed by science, which are the laws of probabilities of the quantum world, is confirmed. Similarly, Schrödinger in the book What is Life stated, new laws to be expected in the organism. What I wish to make clear in this last chapter is, in short, that from all we have learned about the structure of living matter, we must be prepared to find it working in a manner that cannot be reduced to the ordinary laws of physics. The mentioned general genomic rules of probabilities are candidates for the role of such laws. These rules testify that genetic informatics is based on probabilistic principles connected with quantum mechanics and quantum informatics. In general, the discoverable rules indicate that nucleotide sequences of genomic DNAs are stochastic deterministic, that is, those in which stochastics are strongly limited by the presence of regular relationship between the total probabilities of certain groups of N-plates from different N-plate layers. The matrix representations of these probabilistic rules revealed that the matrices of genomic probabilities of the mentioned N-plates are algebraically interrelated each other under different values of N. This interrelation shows a formal connection of families of genomic probability matrices with well-known tensor matrix theory of digital antenna arrays by Professor Slutsar, which gives very advantageous possibilities in the field of digital antenna arrays, also called smart antennas. Antenna arrays have thousands of applications: medical ultrasound scanning, sonar systems, and radio relay stations, avionics, and many others. Modern science connects with antenna arrays future revolutionary changes in computers, biophotonics, and energetics effective using solar energy due to using non-antenna arrays, which can include photonic crystals, liquid clear crystals, etc. Imaging properties of antenna array. Antenna arrays provide such kinds of radiation patterns or beam forming which are unable for a single antenna. Let's list main emergent properties of antenna arrays. Firstly, in a compare with a single antenna, an array of n elements allows to increase approximately n times the directivity and the gain of the antenna, as well as allows to narrow beams. Second, antenna arrays are a unique tool for providing communication noise in unity and extracting a weak signal from a strong noise. Third, phased antenna arrays capable of operating with multi-beam radiation patterns. Given these unique emergent properties of antenna arrays and also structural analogies between tensor matrix theory of antenna arrays and the mentioned genetic rules of genomic probabilities, it can be expected that organisms use genetically inherited bioantenna arrays in their life. This expectation was confirmed by many examples of the structure and functioning of inherited biological systems based on antenna arrays and their energy information waves. Let's show some examples. Considering these examples, one should remember that electrical and vibromechanical oscillations in living bodies are closely connected because many tissues are piezoelectrical: nucleic acids, actin, dentin, tendons, bone, etc. Example 1, complex faceted eyes. Insects and some other invertebrates receive visual information through complex faceted eyes, which serve as bioantenna arrays for receiving electromagnetic waves. Example 2, biological echolocation. Many organisms have an innate ability to echolocation based on directed rays of a wave nature. Due to the mechanisms of echolocation, for example, dolphins and bats are able to recognize the distance, dimensions, and shape of the objects they track, by analogy with how active smart antennas make it possible in technology. A dolphin's body has many hydroacoustic receptors, which form a multi-element broadband hydroacoustic receiving antenna. Example 3, inherited sets of biophoton crystals. Figures. Photonic crystals form heritable species patterns on butterfly wings and peacock feathers. Modern technology actively uses photonic crystals to control the spatial distribution of photon beams. A photonic crystal is a periodic optical nanostructure that affects the motion of photons. It is known that photonic crystals are related to the topic of nano-antenna arrays. Taking into account all these facts, the author has put forward and substantiated the doctrine of energy information evolution based on bioantenna arrays, which explain many facts of physiology. In the end of the brief presentation of the broad topic of genomic probabilities in connection with the Boolean genetic code of athletes and cyclic grade codes, we recall that probability theory itself is closely related to abstract Boolean algebra, being one of its possible interpretations. More precisely, the objects of probability theory is a set of elements that form a normalized Boolean algebra. One can also recall the binary law, all or none, for active biotissues. A nerve cell and a muscle unit give only yes or no answers to the actions of various stimuli. They don't respond to sub-threshold stimuli, but to supra-threshold stimuli. They respond with full amplitude. This is similar to the aberration of computer triggers. Next, collective algebra logical consciousness. Biology is saturated with phenomena of purposeful logical interaction between parts of a whole biosystem. The discovery of the connection between the genetic code system and Boolean functions, Boolean genetic code, etc., provides new approaches to understanding these inherited phenomena. Let me present some examples. The single-cell organism Mixed-Risha paradoxa moves due to 250,000 spiral bacteria Treponema spirochetes located on its surface. The spiral flagella of which twist in a coordinated manner as a single whole, ensuring the appropriate movement in the required direction and speeds due to logical interactions and logical control. Vortex algae are colonial mobile organisms consisting of many flagellated unicellular organisms and purposefully moving due to a logically coordinated beating of the flagella of all cells. In biology, it is believed that multicellular organisms evolved from unicellular organisms through such colonial forms with their inherited logic. Holistic organisms, when solving collective problems, also interact with each other on the basis of innate logical mechanisms, without the need for complex individual learning. Examples: the dance of bees with the transfer of information through the logic of movements. Second, collectivism in an ant colony where each ant has its own specialization, but acts in the interests of the colony, on whose survival its life depends. Multi-million termite communities are endowed with a collective logical consciousness, mega-brain, creating the unthinkable for an individual insect. They build termite mounds up to 13 meters high with a huge system of air ducts and living chambers in order to maintain a certain humidity and temperature necessary for their life. They conduct agriculture, growing mushrooms in termite mounds to feed the colony, etc. It seems that biological self-organization as a whole is based on the inherited logic of interrelations connected with the Boolean algebra of logic, with which the system of genetic coding and the fundamental physiological law of all or none are related. Ancient Indian philosophy, genetic trans, informatics, and chess. The important topic concerns structural parallelism existing between the molecular system of genetic code and the provisions of ancient Indian philosophy. This parallelism allows us to think that at least some of the schemes of ancient Indian philosophy did not arise out of nowhere, but are a reflection of the algebraic organization of the genetic code system. Deep provisions on the connection between ancient Indian philosophy and mathematical logic are contained in the book by Professor Ranjan, Brahma Sutras, A Logical Interpretation. Let us note some of these parallelisms. According to the Vedic cosmology, the universe is mathematical. Molecular genetic informatics also possesses impressive algebraic structure, which are revealed more and more in modern science. The Lord of the manifested world, Brahma, related the universe is four sounds, consisting of two complementary and in the same time opposite pairs binaries, like four nucleotides of binary alphabet. Sculptural images of Brahma convey the idea of four sums in the symbolic form. Brahma always has four heads with four faces turned in the four cardinal directions and four hands. The mythology, religion, and philosophy of the Indians were based on the four sums: Chatur Veda, four Vedas; Chatur Yuga, four Yuga eras; Chatur Varna, four castes, etc. India is considered the birthplace of chess, and the early chess, Chaturanga, considered of four parts and was played by four players. The set of pieces for each contained four pawns and four roll pieces. Its figures were placed in the north, south, east, and west. One of the arguments in favor of the fact that the birthplace of chess is India is the mention in the Vedas and Agni Purana of a plan of 64 cells as a mystical diagram of the Vedas, a model of the world. The chessboard, which carries a cross-like mosaic of 64 white and black cells, represents the harmony of fractal-like constructions, since it contains four cross-shaped quadrants, each of which also contains four cross-shaped subquadrants. The Brahmin priestly caste used a similar 8 and 8 diagrams of 64 cells, which is called Vastu Mandala. As such a fractal, it was taken as a basis for the construction of settlements and temples. It is gratifying for me to know that the current world chess champion, the youngest in history, is Indian Grandmaster Petoukhov Domaraji. Ancient India adored squares, so even their astrological star charts were square. In the fractal-like layout of the 64-cell square, each cell was assigned its own deity, that is, such a set of 64 cells correspond to a whole pantheon of deities. Significant attention in the Vedas and ancient India concepts is given to cyclic processes, including the wheel of samsara, the cyclicity of all life, matter, and existence, is a fundamental belief of most Indian religions. Analogically, the genetic code system is structurally connected with cyclic gray codes to code cyclic physiological processes. Multi-dimensional cyclic gray codes and the genetic chessboard. Along with the one-dimensional gray codes described above, there are multi-dimensional gray codes, primarily gray two-dimensional and three-dimensional codes. They are used in technologies, for example, in quadrature amplitude modulation to transit color signals in the PAL and NTSC television standards. In gray two-dimensional codes, the Hamming distance between adjacent terms, code words, is 2, and in gray three-dimensional codes, the Hamming distance between adjacent terms is 3. Let us return to the genetic Carnot 8 and 8 matrix of 64 triplets, whose cells are enumerated with one-dimensional 6-bit gray code. Great two-dimensional codes divides the set of 64 numbers of this 6-bit gray code into two subsets, one containing all even numbers and the other containing all odd numbers. Accordingly, the set of 64 cells of the genetic Carnot matrix is divided into subsets of 32 cells, in each with even and odd numbers, which is expressed by the white-black even-odd mosaic of the Carnot matrix. See figure. This genetic mosaic matrix, resembling a chessboard, we call the genetic chessboard. The main differences between the genetic chessboard and the ordinary chessboard are as follows. Firstly, all 64 cells of the genetic chessboard are numbered by members of the 6-bit gray code, which introduces the Hamming distance between the cells by their code numbers, and which connects it with the Boolean algebra logically conjugated with the genetic Carnot matrix. Secondly, each of the 64 cells of the genetical chessboard corresponds to one of the 64 triplets of the genetic code. On the genetic chessboard, the sets of 32 black cells and 32 white cells contain completely identical sets of amino acids and stop codons. This further confirms the deep connection of the genetic coding system with cyclic gray codes and genetic Carnot matrices. It turns out that on the genetic chessboard, the classical moves of chess pieces are expressed with cyclic gray codes in their one-dimensional, two-dimensional, and three-dimensional versions. That is, the moves from the initial cell into a new cell, whose 6-bit numeration differs by the Hamming distance 1 or 3 or 2 or 3. Indeed, on the chessboard, all pieces, except the knight, move either vertically or horizontally or diagonally. On the genetic chessboard, the vertical and horizontal moves of pieces are series of transitions to neighboring cells whose 6-bit numbers differ by 1 bit and are therefore described as transitions by the gray one-dimensional code, Hamming distance 1. And the diagonal moves of pieces are series of transitions to neighboring cells whose 6-bit numbers differ by 2 bits and are therefore described as transitions by the gray two-dimensional code, Hamming distance 2. The moves of the knight on the genetic chessboard turn out to correspond to the gray three-dimensional code, since the knight's move always takes it to a cell whose number differs by exactly 3 bits from a number of the original cell. Hamming distance is 3. The roots of the knight of the ordinary chessboard have been the subject of the works of many mathematicians, starting at least in the 18th century. The most famous of these are works of Euler and his followers on the question of whether a knight can go around all 64 cells of a chessboard without stepping on any cell twice. As established by mathematicians, there are 26 trillion such cyclic roots, in which the horse from the last point of the rows can go to the starting point for a cyclic repetition, the entire route an infinite number of times. The speaker believes that chess is linked to genetic archetypes that serves a hidden reason for its popularity all over the world for thousands of years. Is it possible that chess genetics? The genetic chessboard is associated with triplets and amino acids. This allows matching the roots of chess pieces with amino acid sequences in proteins and with triplet sequences in genomes and genes. For example, a particular root of a knight on such a genetic board corresponds to a certain sequence of triplets and amino acids, which can be matched with real genetic sequences. See detail in my preprints. Could there be benefits from developing such a kind of chess genetics? This is an open question. Additional materials of this tutorial are presented in my book, Algebraic Biology, Matrix Genetics, and Genetic Intelligence, co-authored with Professor Matthew He, USA. And in the articles, including my web site. Conclusion. The presented topic, quantum bioinformatics and ancient Indian philosophy, is meaningful and promising for further study leading to new approaches in bioinformatics, quantum computing, artificial intelligence, and biotechnology. Thanks for your attention.